Hello, my name is Zane. I'm a master's student in water resources engineering at the University of Kansas, and I will be talking to you today about my research on the widespread presence of cyanotoxins and taste and odor compounds within benthic algae of human disturbed rivers. For a quick background, cyanobacteria can release many different chemicals or metabolites, some of which are taste and odor compounds and cyanotoxins. In this research, we are studying the specific taste and odor compounds geosmin and MIB, which are expensive to treat out of drinking water, and the cyanotoxins microcystin, anatoxin A, and saxitoxin, which can cause poisoning and even death in higher concentrations. Also, this analysis is focused specifically on benthic algae, which grow attached to a substrate and are less understood compared to their planktonic counterparts, and the variables that promote the production and release of toxins are poorly understood. With this study, we investigate how urbanization impacts concentrations of cyanotoxins and taste and odor compounds and their gene sequence concentrations. The research objectives were to analyze the physicochemical and biological impacts on cyanotoxin and taste and odor compound concentrations and their respective gene sequence concentrations, and to relate land use impacts on water quality and cyanotoxin and taste and odor concentrations. The study site in question for this research include 3 streams in Johnson County, Kansas, just southwest of Kansas City, which have varying land use differences. The most urban has 90% urbanized land use. The second stream exhibits a mixed land use at 62% urban, and the third is more rural and agriculture based at only 21% urban land use. To better understand what causes cyanobacteria to release cyanotoxins, we look at the drivers and factors related to this. Benthic algae thrive in a complex environment of water and substrate interactions, nutrients, light, and many other characteristics. Urbanization can impact these characteristics in several ways, including impacting tree cover, water quality, temperature, stream channel hydrology, and more. From the differences in these factors, we would then expect a range of benthic cyanobacteria toxin dynamics that we could then analyze. The flowchart below this demonstrates our methodology. In each stream, we chose 3 benthic mat sites. We then performed sensor-based and collection-based readings on the benthic mats themselves and in the water. The collected samples are then processed and analyzed, and they're split for multiple analysis, including taxonomic ELISA kit analysis for concentrations and qPCR DNA extraction for the gene sequence concentrations. Finally, linear regression and random forest modeling was performed in order to elucidate relationships between physicochemical, biological, and other data points and the cyanotoxin and taste and odor compound dynamics. For the results on this poster, we've streamlined them to only show microcystin as it had the most detections. However, the conclusions drawn are similar for the other cyanotoxins and taste and odor compounds. First, microcystin was detected in every benthic algae sample. Looking at the microcystin concentration results separated by stream, we can see that except for one site in the most urban stream, most sites demonstrated similar concentrations of microcystin. The second plot here shows the random forest regression using a data set with biological, physicochemical, and land use data points in order to try to predict microcystin concentrations. As seen by the strong R-squared of 0.94, the random forest models outperformed the linear regression models. A Shapley value analysis was performed on the random forest model to determine which predictor variables had the greatest strength in the random forest models developed. From the Shapley value results, we determined that the gene sequence concentrations, the site cyanobacteria concentrations, the site temperature, and the specific conductance were the strongest predictors. Cyanotoxins and taste and odor compounds were prevalent in many algae samples. While urbanization did not show any strong correlations to cyanotoxin or taste and odor concentrations, our results do suggest that any human-derived environmental impact, whether urbanization or ruralization, can lead to the occurrence of cyanotoxins and taste and odor compounds within freshwater benthic mats. Thank you.